to happen. Our chair, Hanbai Marwick, with whom I published several papers promoting drug repurposing, my husband, Alan, who's doing administration, and Claire Dransfield from Action for ME, who's hosting this Zoom call. Thank you to each of you, without whom these workshops would not have happened. Sorry, I'm having problems moving on. There we go. Just some housekeeping. This workshop will be recorded to make it available afterwards, though there will be a slight delay because of holidays. We're here so we can contribute to discussions and listen and respect all contributors' opinions. You may not always agree, and do feel free to comment, but please don't be abusive. As is general policy nowadays, any abusive language will not be tolerated, and in the very unlikely instance of anyone persisting after a warning, they would be removed from the workshop. There are breaks scheduled today, but if you're a person with ME, feel free to take extra breaks as you need. So to the workshop. I'm a person with ME, not a researcher, but I've been involved with attempting to get funding for drug repurposing studies in ME for the past 14 years, as I had a remarkable improvement to a drug. Treatments for ME are a top priority for people with ME. As you can see from this slide of the results of the top 10 research priorities from the 2022 ME CFS James Lind Alliance Priority Setting Partnership, which was voted on by people with ME, their carers and clinicians, probably including people here today. Existing drugs or drug repurposing was voted the second most important question, and the word treatment appeared in 6 other questions in the top 10. Despite this, there have been very few clinical trials of drugs in people with ME. This recent systematic review of drug treatment trials showed that very few studies gave positive results and none were conclusive. Don't worry about the details, as my slides will be available afterwards. Choosing drugs that make a significant difference or a cure will only become easier once the underlying mechanisms of ME are understood. Until that happens, we're looking at drugs to help symptoms. One of the myths which people with ME are told is that there are no treatments for ME. That is untrue. Although there are no cures, there are drugs that help, at least a bit, at least in some people. But different drugs help different people, and sometimes nothing helps. It's hardly surprising people respond differently when most researchers believe ME is a collection of different diseases with the same symptoms, termed heterogeneous. We don't yet know how to predict which drugs work for which person, and that affects how you approach designing a drug clinical trial. Despite that, clinicians are using drugs in clinical practice in several countries. This slide and the next are the drugs recommended by the US ME/CFS Clinician Coalition and the European ME Network of Clinicians, both of which have some very experienced clinician researchers in their midst. So what drugs do people with ME want trialled in the UK? We have a good snapshot from the JLA PSP First survey, which was an open-ended question asking what people would like research to find out. This is the list of drugs that were mentioned and the number of times they were mentioned by people responding to the survey. LDN is quite clearly top of that list. So if we wanted to choose a drug for a clinical trial in the UK, LDN seems a very good first choice. It also agrees with what the US Clinician Coalition recommended in 2019 when they issued a joint statement which ended, "The ME/CFS Clinician Coalition agrees that our first priority is a trial of LDN." As I said, I had a remarkable improvement to a drug, in my case LDN. I published my story as a part of a case series, and these are before and after photos of me to show that drugs can make a difference, at least in some people with ME. I was mainly housebound. I'm in a motorized wheelchair on the left, and I now have a near normal life. I don't want everyone to rush out and start LDN because of what I'm saying. I'm clear that LDN doesn't work for everyone, and some people can get side effects, so they find they cannot continue taking it. There are also other options, treating specific symptoms of ME, such as dizziness from orthostatic intolerance, pain, gut problems, allergies, sleep difficulties, or depression, and trying any drug or alternative treatment before someone has found their baseline level of functioning might mean you were stuck on a drug or expensive alternative therapy when you could have improved anyway. Without clinical trials, it's difficult to know whether improvements are genuine or due to the placebo effect. Even I don't know for sure. This is the final rituximab study showing that for people who had placebo injections, the fatigue score improved by an average of 35%. That improvement is enough to make a real difference to someone's health and ability to look after themselves, and it is a genuine improvement in health. There is little published about LDN as a treatment for ME, and so far no published clinical trials. I've listed here some publications for both ME and long COVID. Because of the placebo effect, retrospective and even prospective open-label studies don't tell us very much. However, there are 2 well-designed trials which have recently started in Vancouver for long COVID and Boston, Massachusetts for ME. For fibromyalgia, there are more publications of higher quality, and we're really lucky to have with us today 2 principal investigators for 2 of the studies, one finished and one still in progress. There have also been more attempts to find the optimum dose of LDN, at least one formal dosing study and one retrospective chart analysis. But it's obvious more work is needed on the optimum dose and how to start people on LDN. So for now, we have to wait before we know if LDN or any other drug is effective for ME and at what dose. The UK medical system is restrictive on what drugs a doctor can prescribe. NICE issues guidance on treatments for different diseases and uses only high-quality clinical trial evidence on drug efficacy before it will recommend a drug is used. So no drugs were included in the latest NICE guidance for ME CFS. There are also potential issues for doctors if they want to prescribe something not recommended by NICE and which is not already licensed for that particular illness. We do know LDN is a very safe drug, but it is not listed in the BNF, though a much higher dose of naltrexone is. This slide is repeated from the first workshop, but I make no apology for that. Over the past 10 years, the UK national funding bodies have funded about £8.5 million of research in ME. One clinical drug trial costs about 2 to 4 million, so that equates to just 2 to 3 clinical drug trials in total. Compare that to Cancer Research UK, which has an annual income of 719 million, and it receives a lot of funding from public funders as well. If health research funding was distributed by disease burden, ME research would receive about £20 million every year. We know from the second workshop that a good platform study of repurposed drugs, adding additional drugs over time as the science or experience suggests, new candidates, costs about £50 million. The particular study we heard about, the Octopus Platform Study for Progressive MS, was funded by the MS Society. Given the lack of funding up to now, we either need a change of heart at government level to make ME research the urgent priority that it really is, or we need to raise £50 million to break out of the current deadlock. Either way, this needs the ME community to come together in a massive effort to change the direction of funding. Are we able to achieve that? I've shown there are a lot of queries about drug repurposing studies, but also they're really needed if drugs that help people with ME are to become freely available on the NHS. So how can this workshop help? We won't settle the difficult questions today, though we will be looking at gaps in current knowledge. The aim is to generate ideas, hopefully spark some collaborations, and work out a route forward which will stimulate clinical research in the UK. In all the discussions we have, can we ask ourselves, what are the next steps we could take and how? Thank you.
Thanks, Monica. That was crystal clear and exactly on time, as always. Monica and I have been working for a long time, and I'm always amazed by how well she is prepared and how wonderfully well everything is executed. So I'm very happy to introduce the next speaker, Dr. Karen Duer-Bruun from Denmark, from Odense Pain Research Group. She recently published a clinical trial of LDN in people with fibromyalgia. Dr. Karen is also a physician who treats people with LDN. Karen, over to you.
Thank you. I'll just start with sharing my screen. And I hope it works. It seems to work. So thank you very much, Monica and Harm, for the invitation to speak at this workshop. I've been working with chronic pain for the last 13 years, and I've just finished my PhD about fibromyalgia and low-dose naltrexone. And I'm not an expert in ME. However, there is a large overlap in some of the core symptoms of ME and fibromyalgia. And before I go into detail with the studies, I'll just briefly introduce the symptoms related to fibromyalgia and which are the currently recommended treatments. Fibromyalgia has recently been recognized as a primary pain disorder, and the core symptoms are widespread pain and widespread tenderness, together with moderate to severe degrees of insomnia, fatigue, and dyscognition. And on top of that, there can be a range of somatic symptoms and emotional symptoms that often vary between patients. Currently, there are a number of evidence-based treatments that are recommended for fibromyalgia. However, no available treatments, pharmacological as well as non-pharmacological, has a general effect in fibromyalgia. And this means that only few patients benefit substantially from these treatments. About 10 years ago, low-dose naltrexone, also named LDN, popped up in the literature as a new treatment for fibromyalgia, and results from these two small clinical trials seemed promising, so it gained hope that LDN could be a new effective treatment. And also about 10 years ago, I met a patient who used it and had benefited very well from it. And soon after, I began using it for my own patients with fibromyalgia who had tried everything else without benefit. And I used the titration approach, titrating patients gradually from 0.75 milligram to a maximum of 4.5 milligram, which was the dose that had been used in the published trials. And my experiences were that LDN was very well tolerated in most patients in these doses, and many patients reported improvement of pain, sleep, fatigue, or dyscognition, and also sometimes a combination of these. So I was really motivated to improve the evidence and to conduct a larger trial with a more robust methodology. So the main subject of my PhD thesis was to investigate if low-dose naltrexone could be a new effective treatment for fibromyalgia pain. But at this early point, I was afraid that the 4.5 milligram might be too high and would lead to too many dropouts. So we planned two clinical trials. First, the dose response study, which was published in Pain Medicine in 2020, and then followed by a randomized placebo-controlled trial, which has recently been published in the Lancet Rheumatology. And I'll just briefly explain the dose response study, but my emphasis will be on telling you about the randomized trial. For the dose response study, we used the up and down method. And in this method, instead of exposing the same patient to many different doses, all participants only receive one dose, but they receive different doses. And the test dose is randomized only in the first patient, and then the response in the first patient determines the dose in the next patient. So if the first patient has a positive response, then the dose goes one step down in the next patient, and if the patient does not respond, then the dose goes one step up. And this continues until the dose has gone up and down 10 times. So we had a predefined dose range from 0.75 milligram to 6 milligram and a predefined dose interval of 0.75 milligram. And you can see here our up and down curve, and based on this curve, it can be estimated which dose will be effective in 50% and 90% of the patients. And you can see our estimates here at the right. However, seen in retrospect, I think there are too many problems with this design to allow for any conclusions about the most appropriate test dose. but the study functioned very well as a feasibility study where we gained experience with higher doses. So it was mainly based on these experiences that we chose to use 6 milligram as a test dose in the randomized trial. The randomized trial, also named the final trial, was a double-blind placebo-controlled trial that was designed and conducted according to the highest standard guidelines. And the participants were women aged 18 to 64 years with fibromyalgia, and they were recruited from all over Denmark. The intervention was naltrexone 6 milligram, but we chose to include a titration phase and allowed for lower doses in case of problems with tolerability. And the comparator was an identically looking placebo tablet. And the primary outcome was the average pain intensity during the last 7 days, which was measured using an item from a validated questionnaire named the Fibromyalgia Impact Questionnaire Revised. The primary outcome was measured using a numeric rating scale ranging from 0 to 10, and we wanted to be able to demonstrate a difference between the groups of 1 point, which corresponds to a large effect size. And to do this, it would require around 100 participants. In addition to our primary pain outcome, we also included some supportive pain outcomes, being the number of patients with a 15%, 30%, and 50% improvement of pain. And here the 30% pain response was of our special interest, as this has been defined as a clinically meaningful improvement for the individual patient. And also these 30% and 50% response rates are used to calculate something called numbers needed to benefit, which is a number that roughly tells how many has to receive a treatment before one person benefits substantially. And especially if a general effect cannot be demonstrated, then the numbers needed to benefit can be used for a risk-benefit assessment where this response rate is held up against the dropout rate. But I'll get back to that later. We also included a lot of secondary outcomes, and I'll not go into detail with all of them, but just highlight that we of course included the 3 core non-pain symptoms that I mentioned in the beginning. However, regarding cognitive dysfunction, we only asked about memory problems using another item from the Fibromyalgia Impact Questionnaire. We ended up including 99 participants that were randomized to treatment with LDN or placebo. And the first 4 weeks was a titration phase, and then the last 8 weeks, the dose remained stable. And the patient-reported outcomes were assessed every 4 weeks. We were delayed by 1 year because of the COVID pandemic, but apart from that, there were no difficulties during the trial, and everything actually went pretty well as scheduled. So after 12 weeks of treatment, we found a pain reduction in the LDN group of -1.3 and a pain reduction in the placebo group of -0.9. And the difference between groups was 0.34 in favor of LDN. And this difference was not statistically significant. As you can see here from the confidence interval, it actually excludes a difference of 1.0, which was the difference we aimed to detect. So our study was negative, and our estimate of the effect pointed to a small effect size. And as you remember, our study was powered to detect a large effect size, and actually it would have required around 600 participants for us to get a significant result with this group difference. But it gets interesting if we look at the trajectories of the pain outcome, because here you can see that the pain intensity goes down in both groups, but you can see that the first 4 weeks there are no difference, and then the curves begin to go more and more apart. So when looking at this curve, it could have been interesting to see if this tendency would have continued if the treatment period had been longer. And here you can see our pain responder indices, which is in my opinion is the most interesting finding from the trial. And as you can see from the risk ratios here, they were all in favor of LDN, but none of them were statistically significant, because in that case, both the lower limit and the upper limit of the confidence interval should be above 1. But especially if we look at the 30% pain improvement, you can see that the lower limit is actually pretty close to 1, and this could be interpreted as a potential statistical significant difference. So unfortunately, our study was likely underpowered to detect this difference as well. But if we just suppose that this estimate was true, it would mean that we could estimate the numbers needed to benefit around to be around 7. And this is actually pretty similar to currently recommended treatments. The patients tolerated the treatment very well, and the dropouts due to adverse events was low in both groups, and with a dropout of 8% in the LDN group and 6% in the placebo group, and numbers needed to harm of 50 could be estimated from our trial, and this is pretty excellent compared to recommended treatments. So regarding the secondary outcomes, I'll just highlight the results regarding fatigue, sleep disturbance, and memory problems. And I must admit that I was surprised that the difference between groups was so small for fatigue and sleep disturbance. But we actually found a large difference regarding improvement of memory problems. However, because we had so many secondary outcomes, this difference did not remain statistically significant after we adjusted the significant level due to these multiple outcomes. So the conclusions that can be drawn from the final study would be that we found no issues regarding safety when using doses up to 6 milligram naltrexone in women with fibromyalgia, and that the final trial actually ruled out a large effect size from treatment with LM on fibromyalgia pain. But we found that LDN might be effective for the treatment of memory problems associated with fibromyalgia. And our data suggests that LDN could be effective for the treatment of pain in some individuals with fibromyalgia. And I'll just comment a bit more on that, because all current recommended treatments have low effect sizes, and therefore their recommendation is based on a risk-benefit assessment. And if we take a look at duloxetine and pregabalin, where the treatment is actually based on high-level evidence, we can see that they both have relatively high numbers needed to benefit and relatively low numbers needed to harm. And if we compare with the estimates we had from the final trial, we could get an impression that they might have a favorable profile compared to these treatments, especially regarding the numbers needed to harm. But of course, this is only an estimate based on 99 participants, and therefore much more LDN studies are needed before we can make an accurate risk-benefit assessment. I generally think that we designed and conducted the RCT pretty well. However, several lessons can be learned for the benefit of future trials. So when looking back now, it seems pretty optimistic to think that we could demonstrate a large effect size when all other treatments have low effect sizes. So what seems to be more interesting is the pain responders, and the study was underpowered to detect a difference in those as well. And as I already mentioned, our pain trajectory suggests that our treatment period might have been too short. And since we planned the final trial, I've gained experience from the clinic with doses up to 9 milligram, and I believe that the test dose might have been insufficient in some of the patients. So my recommendations for future trials would be, firstly, to conduct a new dose response study using a larger dose range, and I'll also suggest to use another method. And in pain efficacy trials, I would suggest to use pain response rates as the primary outcome instead of change between groups. And based on our pain trajectories, I would recommend a longer treatment period than 12 weeks. And I'll highly recommend to include a titration phase and to allow for different doses, as this reflects reality much better. And in our study, I'm convinced that this approach was one of the reasons that we had such a high adherence to the treatment. And finally, it would be very interesting to further investigate a possible effect on memory problems or other aspects of cognitive dysfunction. And I think that this could be especially relevant in ME, where pain is not a dominant symptom. So that was all from me. Thank you very much, everybody, for your attention.
Thank you so much, Karen. That was, like Monica, perfectly on time and a crystal clear presentation. We have some time for discussion after the next speaker, Dr. Juan Luciano from the Universitat Autónoma de Barcelona, Spain, who is currently leading also a clinical trial of LDN in people with fibromyalgia. Juan, please. The floor is yours.
I'm going to share my screen, my presentation. Okay. Okay. Well, first of all, thank you, Monica. Thank you, Han, for inviting me to join this interesting workshop. It's an honor for me to be here. The title of my presentation is a randomized double-blind placebo-controlled phase 3 trial examining the add-on efficacy, causality, and neurobiological effects of LDN in patients with fibromyalgia, the INNOVA study. First of all, let me to introduce my colleagues at the Agora Research Group. We are a multidisciplinary group composed of medical doctors, psychologists, physiotherapists. Our main interest is to study what treatments work for patients in pain, how they work, and the neurobiological effects and mechanisms of these treatments. As my colleague said, the main characteristic of fibromyalgia is widespread pain, while the American College of Rheumatology classification criterion 1990, the most famous classification criterion, focus on 18 points. Patients do not usually report tender points. They usually report generalized pain. But there is also a constellation of additional symptoms like fatigue, cognitive problems, fibro fog, sleep disturbance, affective problems, distress, and so on. One of the main characteristics of fibromyalgia patients is the presence of hyperalgesia and allodynia. That is an increased pain response to noxious stimuli, hyperalgesia, and non-noxious stimuli, allodynia, as you can see in the graph, compared to subjects without fibromyalgia. In our country, I guess that also in Denmark, in United Kingdom, the prevalence of fibromyalgia is very high. In our country, around 1 million people has fibromyalgia, and the economic cost in the form of healthcare visits, medication consumption, days under sick leave, the economic costs are very high. It has been estimated at more than 13 million euros annually. As you know very well, there have been many systematic reviews and meta-analyses in fibromyalgia to study the efficacy of fibromyalgia treatments. They conclude, you can see that pharmacological interventions like serotonin and norepinephrine reuptake inhibitors and pregabalin are more effective than placebo on pain and quality of life, but the relevance of the effects can we put them in question? It's a small effect size, and the side effects are very important. Regarding the non-pharmacological interventions, multi-component therapy, aerobic exercise, cognitive behavior therapy are effective, reducing pain intensity and improving quality of life. But also the effect size are modest in general. There is a colleague in United Kingdom, you probably know him, Dr. Kim Lawson. He published 2 systematic reviews about promising pharmacological treatments for fibromyalgia. And among the list of potential and promising treatments, I want to highlight naltrexone, low-dose naltrexone, as one of the most promising because of the previous crossover trials made by my colleague Jared Younger, firstly at Stanford University and now at University of Alabama at Birmingham. Well, what is LDN and how does it work? It's important for our audience to know how does it work. A low dose is a dose usually considered between 1.5 and 4.5 taken once nightly. And the potential mechanism of action is the next one. LDN taken with this dose between 9 p.m. and 3 a.m. blocks the opioid receptor transiently. Briefly and temporarily blocking the endorphin receptors at night triggers a rebound stimulation of endorphins the following day. And this paradoxical effect has not been seen with higher dosage, the 50 milligram usual dose of naltrexone. But there are other mechanisms of action. The LDN has, as I said, analgesic, anti-inflammatory, neuroprotective properties that are not dependent on opioid receptor antagonists, and that they are related, these effects, to microglia inhibition in the nervous system. What does it mean? It has been recently discovered that naltrexone is a 50-50 mixture of both dextro and levo isomers, and each isomer has a very different and distinct biologic activities. The L isomer is a pure opioid antagonist, okay, and it is a reversible competitive antagonist at mu and kappa and to a lesser extent opioid receptors. But also, naltrexone, the isomer, is an antagonist for certain immune cells, such as toll-like receptors. This antagonist results in decreased cytokine levels, TNF-alpha, and therefore this reduces inflammation. And this immunomodulatory effect is not seen in the doses of 50 to 300 milligrams. Very recent systematic reviews, one of them from Harm and Monica, concluded that there are potential and promising benefits of low-dose naltrexone and that more robust randomized control trials are needed. But one of the main conclusions is that there are no brain imaging studies reporting the effect of how LDN works in patients with fibromyalgia. And the problems are always the same. Small sample sizes, all trials restrict to women, and the risk of bias is usually high. Also, Rupp and colleagues conclude the same. Further high-quality, well-powered randomized control trials are needed to be conducted to establish efficacy, standardization of dosing, and response times. Taking all this background into account, what were the objectives of the INNOVA study that we started 2 years ago, and we are going to finish in October this year? Okay. Our objectives. Please, you can find all the details of our protocol in this paper published in BMA Open 2 years ago. The objectives of the INNOVA study are the next ones: to evaluate the efficacy and safety of LDN in patients with fibromyalgia, to analyze also the cost effectiveness of this drug at 12 months follow-up, to evaluate changes in glutamate levels in brain areas related to pain processing, and to evaluate changes in inflammatory markers, cytokines, measured in serum, to identify other mechanisms of change of LDN. We expect a decrease in glutamate levels in pain-related brain areas. Well, this is the explanation. I want to be brief and concise. There are heightened glutamate levels in pain-related brain areas that have been reported in patients with fibromyalgia, and a putative role of this alteration, and there is a putative role of this alteration in central sensitization that has been proposed by some authors. A chronic low-grade pro-inflammatory status has been reported in patients with fibromyalgia, and this pro-inflammatory status heightens levels of glutamate in brain due to microglia activation. And we know, because of previous studies, that an anti-inflammatory effect of LDN has been previously reported. This is the flow chart of our trial that you probably know, and we allocate finally 100 patients to LDN and placebo, because we couldn't reach the 120 expected number of patients because of budget constraints and because of the medication provided by Accord Healthcare had an expiry date that we had to respect, and we had to finish the inclusion of patients last October. We perform a clinical baseline assessment, then we perform a 3 months assessment, a 6 months assessment, and a 12 months assessment. But also we collect some biomarkers we collect blood at baseline and at 3 months follow-up. And we also make scans at baseline and at 3 months follow-up, but not in all patients, only in a subsample, 50% of patients in each study. Okay. This is the long list of selection criteria. I'm not going to read the list because it's very long, but well, the usual inclusion criteria, no, that other studies have reported. To have primary diagnosis, to be between 18 and 75 years old, to have at least moderate severe pain, 4 out of 10 at least, to understand Spanish language and provide consent. And there is also a long list of inclusion and exclusion criteria for the biomarkers of study embedded within the randomized control trial. Here you have in the screen a table with our primary outcome measure and the secondary outcome measures. The primary outcome measure is pain intensity. It's a numeric rating scale administered at baseline, at post-treatment, that is at 3 months follow-up, 6 months and 12 months. And there is a list of secondary outcomes, the Fibromyalgia Impact Questionnaire, the DAS-21 for measuring anxiety, depression, and stress, the MISTI for measuring subjective cognitive impairment, the WHODAS for measuring disability, the CAT-7 for measuring worry or general anxiety, and there are also additional measures can provide additional interesting information like the client and service receipt inventory to know the medication consumption, the use of healthcare services, and so on. We also measure the impression of change. And this is very important and very relevant, that, and I can focus on it later. All patients in the trial use an app, the pain monitor app. And some days before beginning the trial, and along 3 months, they answer a short battery of questions each day. The items are about pain intensity, fatigue, catastrophizing, because we want to know the evolution or the progress of each individual in the trial day by day. Because then, with this analysis and employing a sophisticated methodology, we can know the evolution of each individual and to know, or we can characterize who are the patients that respond to therapy, who are the ones that do not respond to therapy. Well, regarding the magnetic resonance imaging, our main test is the spectroscopy to identify to measure the glutamate and glutamine levels and other metabolites. And these are the locations: posterior insula, posterior cingulate cortex, ventrolateral prefrontal cortex. And we also collect blood samples for evaluating some important biomarkers related to fibromyalgia, EL6, EL8, EL17, EL4, EL10, and high-sensitive reactive protein. Here you have some of data till now, the retention rate, and the dropouts. At 3 months, the follow-up, we have a retention of 83%, more or less. At 6 months, 73%. And at 12 months, listing into account that we are currently evaluating patients. We haven't finished. We'll finish next October. And we calculate that we'll have a retention rate of 60-something, 65, 67%, more or less. So we have more dropouts than the Danish study. Regarding the magnetic resonance imaging and the blood samples at baseline, we collect blood from 39 individuals, and at 3 months from 32. And we did the scans in 45 individuals at baseline and in 33 individuals at 3 months. We expect to collect data from more individuals, but it has been impossible to find time slots in the scan, logistic difficulties in a very intensive evaluation that includes this trial. These are the reports adverse events. Till now, we have found 110 reports adverse events, and the most frequently reported symptoms are rash, dizziness, vivid dreams, headache. the common symptoms or side effects usually reported in the literature. And today we have 5 serious adverse events, but all of them seem unrelated to the consumption of LDN or placebo. In this table, you have a comparison between the Innova and the final trial. Please correct me later, Karin, if I am wrong. We use in this trial a dose of 4.5, the most common dose in the previous studies. They use 6 milligram dose. Our last evaluation of all patients will be at 12 months follow-up. Their last evaluation at 3 months. We both measure pain intensity. With a numeric rating scale, they use the pick R item of pain. We have a similar number of participants. We collect blood tests. They do blood analysis we use blood analysis with biobank samples. We have found, I think, more adverse events than them. We have had, I think, more dropouts. I wanted to do these brief comparisons before ending my presentation. And well, this is what I wanted to say today. Thank you.
Thank you, Juan. That was also exactly on time and very clear. So I think now we have roughly about 20 minutes for discussion. I think, Alan, is there anyone who's already flagged that they wanted to respond in the chat? I think we probably can open it by asking people to raise their hand if they want to respond or have questions, comments.
I think all the comments in the chat have been answered, unless anyone wishes to go back to them. So I think you're right. General comment. Right.
So it's open for discussion. 2 studies on LDN, 1 ongoing, 1 finished. Anyone with a reaction? So I see, why was it mainly women in the trials? That's the first one from Dawn. Who would like to respond? Juan or Karen?
Well, that's a good question. We have a problem, and I don't know if they have the same problem in Denmark. Men don't want to participate in trials. They are reluctant to participate. It's like they feel ashamed to confess that they have fibromyalgia, or they don't want to participate. I don't know. I think it's a I don't know, Karin, if you think the same. It's very difficult to recruit men in trials. Very difficult.
Yeah, I agree. We can see that in clinical populations, there are not many men who are diagnosed with fibromyalgia. We know that from population studies that there are a lot of men with fibromyalgia, but they are not diagnosed. So it's difficult for us to identify them. So we're afraid that maybe if we could only recruit 5 or 10 males or men, it would just pollute our our results. But also part of my PhD was a diagnostic accuracy study, and we found out that the new diagnostic criteria for fibromyalgia actually is pretty good at identifying men. So in the future, I think that we could have identified them ourselves instead.
Thanks. I see 2 hands. Jess was first. Go for it, Jess.
Oh, hello there. I was just wondering, particularly, thank you so much for 2 really interesting presentations. I was just wondering, Karen in particular, do you have clinical experience of using the low-dose naltrexone bupropion combination, which is available in England, in the United Kingdom, in the BNF? I only ask because we use bupropion sometimes to treat ADHD, and there's been evidence for some of the ADHD medicines for an effect in ME/CFS.
No, I have no experience with that.
Okay.
Juan, do you want to respond?
Okay.
Sorry, Jess, no clarity for you about this.
okay.
Jess will speak later, so you can come back to that if you want to in your own talk. I think the next hand is Matt. Go ahead, Matt.
You're muted, Matt. We can't hear you.
You have to unmute, Matt. Otherwise,
Right, let's
yeah.
try again. Sorry. Sorry. Hi, my name's Matt Rowley. I was only diagnosed with ME/CFS back in December time. So I'm still quite new to having ME and the symptoms that come with it and understanding it. But one thing I did realise, as really triggered by what you're talking about, men and trials, I suspect that ME might be underreported in men and actually confused with mental health issues, because I think most men, if they go to the doctors and they say, I'm feeling tired, I'm feeling stressed, I'm feeling this, I'm feeling that, they will likely go down the route of, oh, that must be a mental health issue, because it could be as serious as suicide ideation or anything like that, whereas actually it could probably just be hormones and a lot of other things that go on, I guess, with ME/CFS. And I think in particular it's I think it's quite a classic situation for men, and please correct me if I'm wrong, anyone, but classic situation for men to say, oh well, I'll just tough it out, I'll try harder, work harder, get accused of being lazy or not being very effective, so it's okay, I'll just try harder, and then push through and actually make the symptoms worse and worse and worse. So there could be I think an area to suggest that because a lot of GP practices aren't particularly good at identifying ME/CFS. I mean, I've been going with symptoms for probably 10 years to various doctors, and I never considered ME/CFS, didn't even really know what it was until December. And I'm just suspecting that I think there's possibly an underserved group in men where they've maybe misdiagnosed, and maybe instead of maybe it still needs mental health support or whatever, but I still think there's a gap potentially in there, because I still look back and I went and saw a therapist for a few months and I'm just like, I'm absolutely fine, I just got no motivation and I just get really tired quickly, and I just thought it was all I just thought it was all in my head. And then once I started getting the guidance on how to do it through the Edinburgh's pain and CFS clinic, I was like, okay, I know how to pace now. I know how to what to do, and I know strategies. And guess what? I'm not having to sleep twice a day anymore just to get through the day and all those sorts of things. So I don't know. There could be, in terms of groups, there might be other people out there which just aren't actually being diagnosed, as opposed to it just being about women not coming forward for trials. But I'm not sure. I just thought I'd throw that in as a personal experience.
I think that's very helpful, Matt. It's really good that you have told us about this. I think it's food for thought, as Jess says. I think there's a comment from Monica. Go ahead, Monica. Are you on mute, Monica? Or
Sorry,
You're muted, Monica.
I think I'm okay now. Going back to the two studies, Karen, given that you had negative results, can you now hone down to what would be a good primary outcome measure if you had to re-choose? And same for Joanne, is there any kind of reflection of good outcome, a primary outcome measure that would maybe be a bit more sensitive?
Well, actually, you can say that the outcome that we measured that was most promising was actually the memory problems. That was where we found the largest difference between groups. And of course, it could be interesting to dig more into that, as we only measured memory problems, but there are many other aspects of cognitive dysfunction. So this could be very interesting to go into to measure a primary outcome that way. But I don't think that we should give up on the pain outcome as well, because as I highlighted, it seems that LDN is probably at least just as effective as current recommended treatments, and it seems that it's tolerated better. So I think that I'm hoping for more trials so we can allowing for a meta-analysis, and I think that we will get the significance then.
Always looking for significance. Thank you. Sarah has her hand up. Go ahead, Sarah.
Thank you. Just as a general comment, it's rather refreshing to see an area of trials that are being done on women and then applied to men, whether that's appropriate or not, rather than the very long history of it being the other way around. But my question is to Juan. A 30% dropout rate is pretty high, or it certainly is in the trials I was doing in my previous life. And I wondered if you're able to have any insight as to what was causing those dropout rates and what one might be able to do to reduce that in future work.
Yeah, that's a good question. And I don't know if I have a good answer for you. I think that when they leave the trial, they usually say that they don't notice something different after taking the medication for several days, 1 month or 2 months. And if they want to discard one of the tablets they are taking, they discard low-dose naltrexone because they feel that it's pointless to take something that is not making an effect.
Okay.
It's not making a difference.
That's fair enough. So it wasn't, given that it's a fluctuating condition, it wasn't that they were having flare-ups for other causes, or they were overtaken by other events, as it were, which is a very big issue for ME research.
I'm not sure of that. We have to discuss what has happened, because yes, the number of dropouts is too high, and at the end of the trial, this can be 35 or even 40%. But taking into account that we are following the patients for a long time. They get tired. They don't want to come back to the hospital. Some of them have very complex lives. And it's difficult to retain them in the trial.
Yeah, of course.
Yeah, thank you.
Thank you, Sarah. Good question. I think Jess had her hand up again. Go ahead, Jess.
Hello. I just wanted to pick up both to Karen and Juan, Monica's question about roughly how much these trials cost. This is because Monica and I have been trying to put in proposals for our own. But I also really wanted to know from Karen, I mean, it seems that the clinical experience is that LDN works for some people. And it would be really interesting to know for whom it works and why. And I wondered, particularly with Karen's data, do you have any other measures, blood samples or, physiological indices that would suggest, even in an underpowered way, whether there is a signal as to who is responding and who isn't? I know that that wasn't the focus of your study, but I just, I wondered if that was something that was interrogatable from your data.
The costs and the signal. Okay, we'll ask Karen and Juan. That might be sensitive, but could you give us a little bit of an idea of how much a study like the one you're doing has costed, and did you find something of a signal?
That's very difficult to say, because it's it's an investigator-initiated trial, so it was my salary as a PhD student, and then there was some operating expenses. And I guess that the operating expenses amounted up to about, I have to calculate from kroners to euro, but I guess about 100,000 euro, I'll guess, the operating expenses. And then my salary on top. So, yeah.
So that's really helpful.
Juan, can you say something about the budget for your project? Roughly?
Yes, of course. We got funding from the Institute of Health Carlos III, the main funding body of biomedical research in Spain. And we got a budget of 270,000 euros for the trial. That for Spain is very generous, because research is underfunded here. And we got surprises because we got the 100% of the budget we present, because they gave a very high score to the trial, to the proposal, and we got surprised because they didn't cut anything. So,
all right. So I think that's an answer to your question, Jess. I think, yeah, we'll come back to the signal. If something comes up, Karen or Juan, and you have an idea of how to answer that, please let us know.
Yeah, we are actually working now on looking into if we can identify a certain type of responder. So that's some exploratory analysis, but we are just in the planning phase. And we also haven't analyzed our blood samples yet. So we have to find out what we are going to analyze. And we have thought about analyzing these endorphin levels and also cytokines, but also there are some clear markers that can be measured in blood. So that's what the idea is, that we have to be to know more about the mechanisms. So
thank you so much. I see a new hand. Monica, do you want to go first, or can we get Ewan in first?
No, go Ewan first.
Ewan, go for it.
Okay, just going back to Matt's point, I think that raises the issue with the different ratios that we see having been reported, over many years of, from 2:1 to up to 4:1 different sex ratios, and the questions that raises, and that is also part of the accuracy of that diagnosis issue, which we have to really try and address, and which is almost impossible in small trials. Unless trials can be joined together more closely and they use sort of quite common protocols and do a sort of double-arm trial, double-blinded within the researchers, where some people are looking at the overall, the general picture, and other people are looking much more closely at the accuracy of individual caseness and the results that relate to that. Then, we really are sort of always going to be sort of just getting a wee bit further and a wee bit further, and lots more to discuss at research level, but not much to actually translate to treatment usefully. Okay.
Thanks, Ewan. That's very helpful. Yeah, I agree. There's another side to this, and that's the diagnostic side. Anything else you want to add, Ewan, or can we go to Monica? All right, Monica, we have 2 minutes.
Quick then. Karen, when I first was talking to you, you were very resistant to using 6 milligrams. You were saying people in clinic, you use lower doses. You're now saying higher doses. Tell us what's happened. What are your now thoughts on this?
Well, what happened is that I got experience from the clinic. So when I planned the trials, my experience was only with 4.5 milligram, and I was very nervous to do this dose response study where we had chosen to go up to 6 milligram, and I was surprised how well it went. Actually, we did not titrate, and we almost had fewer side effects in the high doses. So then I was just convinced that, okay, this should not be a problem. And then I started using in the clinic and got confident that this would work. And then, I hear from people who are not in my care that somebody had benefited from even higher doses, and then we tried to go higher up and find out that some need higher doses than that as well. So I have not tried doses over 9 milligrams myself, but I think that we don't know enough about this dosing range yet.
You mentioned that. Ewan, your hand is still up. Is there an additional point you want to make? All right, thanks. Okay, guys, I think we're just exactly on time. It's 2 o'clock according to the schedule that Monica showed. This was a very nice first bit. I thought it was very interesting. I think we now are scheduled to have a 10-minute break, and then Professor Simon Carter hopefully will join us. So I would suggest you just keep your line open or or come back in 10 minutes. Anything further, Monica, about the housekeeping?
No, come back in 10 minutes time, please. Yes, thank you.
Right on. Thanks. Hi, everyone. I think we're waiting for Professor Simon Carding to log in.
I'm here.
Oh, you're here. Great. Wonderful. That's good news. I see so many little screens that I'm a bit confused. Wonderful. Well, just start. Simon is a research program leader at the Quadram Institute and professor of mucosal immunology at the Norwich Medical School at UEA Norwich, leading two clinical trials in people with ME CFS, the Light Me Up trial and the Restore Me trial. So it's very good news that you're here for this second bit of our workshop on treatments for ME CFS. So over to you, Simon.
Thank you. So I assume you can see my slides. First slide. Great.
Yeah,
thanks, Alan.
we can see them.
Okay, so I'm going to talk about personal experiences in undertaking research in ME and also about our experiences of trying to undertake clinical trials in ME, hence the title of my talk. So with the support of the Invest in ME charity, we have the aim of developing the Quadram Institute as a centre of excellence for ME research. And this includes, so the goal is really with the Centre of Excellence is to develop new evidence-based interventions to treat ME patients. Our fundamental research, which I'm not going to talk about today, concerns of the viruses, viral origin of ME, CFS, autoimmunity, and immune senescence. And then our translation research is clinical trials, which, as Harm said, I'll be talking about the two trials listed there. We are not working alone. We are part of the European ME research group, which comprises about 10 different researchers in 10 different countries. And we've recently formed a Young Emerge, which is for early career researchers to try and advance and promote a career in ME research. And then finally, we're involved in advocacy. We're contributing to discussions with government, both in the UK and in the USA, to develop or increase the funding portfolio for ME research. And in particular in the UK, we're working with UK research, UKRI, to try and again increase the funding that's provided to ME research. So our research focuses on the gut and the possibility that the gut microbiome is a contributing factor or a trigger of ME CFS research. A lot of research focused on the human microbiome now and linking changes in the gut microbiome, so-called dysbiosis, to a whole variety of diseases, and maybe in fact every known human disease as well. So we're particularly interested in how the gut microbiome may contribute to ME CFS. There is quite a lot of evidence for implicating the involvement of the gut and the gut microbiome, and I've listed some of these here on the right-hand side of the slide. More recently, there's been several studies looking at the bacterial component of the microbiome, showing that there are quite distinctive changes that are occurring in the microbiome that may be contributing to the disease pathogenesis. And right at the very bottom there is evidence now that if you can manipulate the microbiome in the gut, then that can be beneficial to at least some ME CFS patients. Okay, so these are some of the challenges that we have faced in trying to do studies, and we are trying to focus primarily on diseases with patients with severe ME. So regarding sort of more patient aspect challenges that we faced is really trying to identify the demographics, the diagnosis, the accuracy of the diagnosis, the case definition that's used, gender and age, and then the onset and duration, how long are the symptoms being manifest, and the severity, etc. An important aspect of our research is defining a control group. Because our interest in the microbiome, we've tried to focus this on individuals that are healthy that live in the same household as the patient in order to control for key environmental factors that contribute to changes in the microbiome, in particularly diet and the living environment. And then, of course, with trying to obtain samples, that's complicated as well, because severe and very severe patients can't obviously come out to us, so we have to visit them. We have confronted issues with the NHS being, and staff in the NHS being willing to support us in this. And again, where do we source these patients from? We've had to use GP clinics, hospitals, homes, everywhere. We've had a variety of means of trying to recruit patients. And then, of course, when do you sample? Ideally, you'd like longitudinal sampling, but that's impractical in many cases. So do you pick patients with active disease, inactive disease, and what time of day do you collect those samples from? These are all potential confounders in the research. And then, of course, what are the samples you're trying to obtain, and how invasive, how disruptive is that to the patient? And of course, the major confounders is we don't really have any biomarkers on which to base the disease or change in the disease severity or duration. Practical constraints relate to ethics. This in the UK can be very drawn out and bureaucratic. It can take months to go through the process to obtain an ethical approval. And then we have to deal sometimes with the MHRA. And again, this being a government agency, it never works very efficiently or proactively. So again, we've significant delays there. And because of that, we have issues with the timing. We can lose patients throughout this process just because of the duration it takes before we can actually start. So we do have to cope with withdrawals. And then something that's not been mentioned, I don't think very much at all, is the legal aspect. So we need insurance, medical insurance for these. And in many cases, we have to bear the cost of insurance for this. And then staffing is also a problem because there aren't really that large pool of people trained that are knowledgeable about ME. So it's not an optimal career option. So it's very difficult to recruit knowledgeable staff. So again, we have to train people. That's another delay. And then, of course, money. Money is a significant constraint. And all of our studies are funded through charitable donations or from patient donations. We've not been able to attract any significant funding from government agencies. And we have relied heavily on these two centres for our patients. So St Helier Hospital, chronic fatigue facility there. And also more recently from the East Coast Community Healthcare, which is based in Suffolk, which has proven to be useful to us in recruiting patients. The question about clinical trials is that they are valuable. Success or failure is really not the primary criteria in many cases, because we can learn an awful lot about the disease from undertaking clinical trials. And therefore, we put a lot of time and effort into developing trials, the photobiomodulation trial and the MRT trial, the two examples of what we're currently working on. I would like to say the key component of our clinical trial design is developing patient and public involvement groups. And this is proving to be essential for making applications, not just to ethical review boards, but also to UK funders. And so we have two types of PPI groups at the Quadram. One is an overall one, which contributes to all human studies. And these consist of team members within the Quadram Institute, as well as members of the public. And we can source a lot of patients or subjects for these recruitments through our volunteer database. The website is there. And then we have an ME special PPI group, which is chaired by an ME patient, a member of QIB. And this group has proved invaluable in helping us design clinical trials, but also review a lot of the outcome measure tools that we're developing, and they provide very useful feedback for this. And one thing we didn't consider was actually site access. How would patients get from the car park to the clinical research facility in the building? So that's been very helpful. We generate videos to help inform patients so they can manage and assess what it's going to take to contribute to the project. So this is the photobiomodulation PBM therapy that we're trying out. And involves low light therapy, which has proven effective for pain, inflammation, wound regeneration, for regenerating bones and tendons. And it's interesting in the context of ME because it targets mitochondria. and particularly the electron transport chain, which is the main mechanism by which ATP, the main source of energy for the body, is generated. So the idea is by irradiating the body with light of a particular wavelength, we're able to essentially reboot the mitochondria, re-energize it to improve energy outcomes. So we're interested in testing whether or not the red light therapy might actually be useful or relevant to ME. So we've undertaken a trial to assess the acceptability and use of this. And we've also incorporated these objective outcome measures. So we're asking participants to wear accelerometers. These are wrist-worn devices which can monitor body position, sleep, as well as physical activity, and then remote tools for assessing cognitive function. And recently we've developed FunCap, which some of you may be aware of already, which I'll mention a bit later on the next slide. So this is a feasibility trial. It's a relatively small one, only 10 participants. We've recruited through websites and social media, and the eligibility criteria is kept broad to try and be as inclusive as we possibly can. And the faces of the people on the slide also indicate the people that are contributing to the study. So we have specialists in red light therapy, Professor Glenn Jeffries, cognitive function, Michael Hornberger, Christian Sonnenberg, who's helped with the FunCap as well. And then Andy Atkins, who's an exercise physiology specialist. So this trial is currently underway. We're about halfway through it. Now, the remote aspects include the use of Mantle, which is a remote research management platform through which we obtain consent. We can do online assessments as well, and we can send automatic reminders to patients as to what they should be doing, when and how. And this is designed specifically to try and reach the most severely affected patients, because we're not requiring them to visit us, and we can mail out the devices, the lamps, and everything that we simply mail back. So it has minimal impact on patients being accessible to the study. This is FunCap, which we've just now published, and this is different to most conventional questionnaires that are used in clinical trials, which generally do not take into account the consequences of doing an activity. FunCap is designed to determine what the impact of undertaking a specific activity is on your ability to do other things. And we have a quantitative scale here, which is indicated on the right-hand side of the slide, ranging from 0, when I do this, I cannot do anything else, to unproblematic, and it does not affect my ability to affect other activities. So FunCap is already being adopted for a couple of clinical trials that we're aware of in the US, and also, I believe, a clinical trial that's about to start in Sweden. And we are using this as part of both Light Me Up and Restore Me. So the challenges we've had with Light Me Up is legal insurance. This has cost over £1,000 for us to obtain insurance policy for this. It's a first of a kind study, so this raises several issues. Patient demographics again, reliance on self-diagnosis, ethics, the assessment of feasibility, power calculations are sort of out the window on this one, because it is just simply a feasibility trial. The online tools is the first time they're going to be tested, and they're really, one of them at least does not come with a manual, so we've had to develop our own guidance for patients to use. Then the equipment, and the Royal Mail in the UK has issues about mailing things with batteries in, so the accelerometers are categorized as dangerous goods, which raises logistical issues for us as well. And then patient compliance and patient completion. Thankfully, so far, we've not had any patients drop out of the study. Very quickly, finally, Restore Me, phase 2B clinical trial to assess whether or not fecal microbiota transplantation can restore a healthy microbiome in patients to improve their physical and mental health. This is based on work carried out by this individual on the right-hand side here, Professor Thomas Barodi, who published a study now, probably about more than 12 years ago, with 60 mild severe ME CFS patients, 52 had irritable bowel syndrome. He did an FMT, which was a transcolonic plus a rectal infusion. 42 responded, improved sleep and fatigue. 58% were symptom-free, 15 to 20 years follow-up, and in the vast majority, their GI symptoms were fully resolved. So that was the base on which we decided or justified undertaking the Restore Me trial. This is a snapshot of what the trial is. It's a single-site randomized, double-blinded, placebo-controlled study. We'll be using capsules to deliver the donor samples, and we're interested in testing both the safety and the efficacy. And this will be the first phase 2b clinical trial to assess efficacy of FMT in ME anywhere. And the first trial we believe to use objective outcome measures, which includes FUNCAP, the neuron assessments, the online cognitive functions, physical activity. All the work I've described, well, haven't really covered in terms of the fundamental research, but they've been involved in the clinical trials of 4 PhD students funded by Invest in ME Research and by the University of East Anglia, with some additional support from Solve ME and ME Research UK. And our collaborators there at the top, Amalak Bansal, who's helped us with patient recruitment, and Tom Wildman, who's a virologist and a colleague of mine at UEA. And with that, I'll stop and thank you all, and I'm happy to take any questions.
Thank you so much. That was brilliant, really interesting, light and microbiome. I think we're planned to have the questions after Jess's talk, but if there are any urgent things that people want to raise, that can be new. I'm not sure, Simon, that you can stay around until after that.
Yeah, I think I should be okay, Hom.
Oh, great. Okay, Jess, can I invite you to do your presentation first, and then we'll have some room for discussion?
Yes, of course. I'm just going to share my screen. Slideshow, play from start. Okay, hello everyone. Thank you so much to Monica for organising these invaluable workshops. It really is a privilege to hear what is priorities for people living with ME, and also what are findings from research colleagues. So I'm going to talk about some feedback from our public engagement events about trying to plan repurposing drug trials for ME CFS. My name is Dr. Jessica Eccles. I work with harm at Brighton and Sussex Medical School, where I'm a reader in brain body medicine at the Department of Neuroscience. I am, for my sins, a psychiatrist, but I hope to explain to you how I got to this point of being interested in drug repurposing trials. And I also lead something called the Neurodivergent Brain Body Clinic in Sussex Partnership Trust, whereas I work as a neurodevelopmental psychiatrist. So I'm just going to give you a little bit of a background to the type of work that we've been doing at BSMS and the outcome of our patient and public involvement. And it really ties in very well with the talks from Juan and Karen. So as I said, I'm a psychiatrist. I do not believe that ME CFS or fibromyalgia or long COVID are psychiatric or functional conditions. I got involved in brain body research through a condition called hypermobility, which some of you may be aware. Hypermobility is associated with orthostatic intolerance, postural tachycardia syndrome. And there are a lot of suggestions about autoimmunity and inflammatory difficulties in this condition as well. And in the course of our work, where we were looking at orthostatic intolerance, we noticed the overlap between hypermobility and fibromyalgia. And this led to further work looking at the overlap between fibromyalgia and ME CFS, and ultimately long COVID. So we were lucky to be funded by Versus Arthritis and Action for ME to do a study of the biological mechanisms of chronic pain and fatigue in fibromyalgia and ME CFS, basically looking to see whether 2 different types of challenges, this is not a clinical trial, this is an experimental medicine study, whether 2 different challenges exacerbated pain and fatigue in patients. One is a autonomic challenge, which provokes orthostatic intolerance, which you can do by putting patients on a tilt table. And the other was a inflammatory challenge, which is giving people typhoid vaccination, which is a mild stimulus to the immune system. So we brought patients in, we put them on the tilt table. We took some basic blood tests, And we then brought them to the brain imaging center, where people attended twice in a randomized, double-blind, crossover fashion. So all participants attended 2 visits, where they at one stage had a typhoid vaccination, and at the other stage they had a saline injection. And we took brain scans and cytokines and various other markers. Now, the interesting findings is that we found that of the patients, most of the fibromyalgia patients, I mean, more than 90% of the fibromyalgia patients met diagnostic criteria for ME/CFS, and more than 90% of the ME/CFS patients met diagnostic criteria for fibromyalgia. So there certainly seems to be a lot of symptom overlap. And as Monica mentioned, we're probably looking at a condition that has got many symptoms, but different subgroups within it. We showed that at baseline, we also showed that of the patients with fibromyalgia and ME/CFS, the vast majority, over 80% of them, had met diagnostic criteria for symptomatic joint hypermobility, which is there's a robust diagnostic criteria involving a physical examination, which determines the presence of joint hypermobility. So we showed that people had, that patients had high levels of baseline inflammatory markers, ESR and CRP, which are routine blood tests that can easily be done in GP surgery, and that these levels in patients were basically at what is considered the cutoff of reference, but in controls they were almost non-existent. We showed that the inflammatory challenge induced pain and fatigue in relationship to how much the immune system responded, particularly if you had a subtype of hypermobility called arachnodactyly, which seems to be associated with autoimmune conditions. We showed that autonomic induced pain was related to the number of connective tissue features in hypermobility, and that autonomic induced fatigue was related to the baseline levels of inflammation. And interestingly, in our fibromyalgia systematic review, we found that one of the LDN studies showed that those who responded most to LDN had higher levels of baseline ESR. And Juan showed us about the immunomodulatory properties of LDN. We've also recently published work showing that if you have joint hypermobility, you're 30% less likely to recover from COVID-19 and develop long COVID. And we also have work in preparation showing that the brain responses to the inflammatory challenge are related to the number of hypermobility features. So we also, and this tied in with the 2 of the previous talks, we did a study of gene expression, so not genetics, but the proteins being produced from the genes in a cell at any one time. And we found that at rest, the patients with ME/CFS and fibromyalgia had differences in mitochondrial metabolism. And after the inflammatory challenge, Juan mentioned the NF-kappa beta pathway, we also found results there. I just want to say that was a huge piece of work, and thank you to all involved. We also have, and you might think this is a bit left field, but we have a paper coming out in July, so the end of July, in BMJ Open, in over 6,000 young people, showing that ADHD and autistic traits in young people are associated with higher levels of inflammation at age 9, and that this relationship is associated with presence of fatigue at age 18. So this is just food for thought. We have proposed, actually, Harm and Monica, our trial is pretty similar, the one that we tried to get funded by the NIHR, to the INNOVA study, in that we were looking to do a subgroup of brain imaging and blood tests. This was not funded, we think, because of cost constraints. And it's becoming increasingly clear, especially from what Karen was saying and what the literature suggests, is that treatments like low-dose naltrexone are a priority, as evidenced by the James Lind Alliance. And they do seem to work in some people. And the question really is, do we need another trial showing whether LDN works or not? Or rather, do we need a trial showing who it works for and why, so that we can target personalized treatments? And that harks back to what Monica was saying about the subgroup element of ME/CFS. So I say here ME/CFS plus in that we are considering our inclusion criteria to include ME/CFS, fibromyalgia and long COVID because of the huge overlap between these conditions. It seems to be somewhat potluck which one you get diagnosed with. So we've been involving PPI right from the very beginning. That's public and patient involvement. And just as James Lind had said, this question is important to those with lived experience. What we've been really trying to find out is how practical and feasible is the research, particularly around what sort of outcome measures are meaningful. So the important thing is to ask people with ME and other energy limiting conditions, and also really to remember spoon theory, which I've forgotten the FUNCAP, which is basically how much will this cost you to do each individual thing in terms of your future energy accounting. So we know that these conditions are poorly understood, and that because the mechanisms are poorly understood, that the conditions are stigmatized and often relegated to the sidelines, and that our PPI shows that really we need to understand the biology and mechanisms of treatment, even if they don't work for all, in order to improve understanding, reduce stigma, and improve access to possible interventions. So we asked participants what things would make it easier, potential participants, our PPI group, what would make it easier to participate in a study of pain and fatigue. And there was strong appetite for the opportunity to do at least part of the trial protocol online. And we've been in discussions with people about how we could go out to people's homes. The ideal facility, and I wonder what Quadrum may have to offer in this, is a residential trial in order that you could actually go and stay. But this could be problematic for carers and parents. People were, they're understandably concerned about childcare, traveller, and energy limiting. And the really important thing to know in advance what is expected of participants. So the two studies that were presented today, the final study and the INOVA study, were both placebo-controlled. So half of the people received a sugar pill. We were concerned when we were setting up our trial design that this seemed slightly unethical and would perhaps be a barrier to people taking part. So we were thinking about a crossover design, which is where everyone acts as their own controls. So it has 12 weeks of treatment and then 12 weeks washout period and then 12 weeks of placebo. But what we found is that the vast majority, and we had about 50 or 60 participants at our public engagement event, said that they would consider taking part in a placebo-controlled study because they felt that it was important to add to the evidence. And people were also keen to take part in a crossover study. We also wondered, it's about the burden towards patients. Would people be willing to have blood tests? Everyone was. Most people would be willing to have skin biopsies, and everyone would be willing to have a brain scan. So one of the further considerations that came up in terms of thinking about things were how to promote diversity, particularly ethnic diversity, and the inclusion of more men. The possibility that people would sign up in order to access additional clinical tests. So if we're offering brain scans and blood tests, and basically, what happens if we show that LDN is successful for certain people, how would they maintain ongoing access to the treatment after the trial? And these are important ethical considerations. People also wondered in this ME/CFS plus arena about people with hypermobility and EDS, and also how frequently visits were possible and how to include severely affected participants. So I just wanted to say what we were thinking is, as I mentioned before, that we would like to consider a trial of who does low-dose naltrexone work for and why and how. We're also considering possibly setting up a cohort and doing a trial within a cohort. And I think it's really encouraging to see other people's research in terms of the INNOVATE and final studies, which makes it really important, I think, about this primary outcome measure, because the primary outcome measure is the thing on which the power is determined, and the sample size and the money all is associated with the power. So it's working out what is the most meaningful primary outcome.
Thanks,
and it's a
Jess.
huge piece of work. So that's it. I just want to say a huge thanks to our charity support partners, so the funders, FibroDuck, Action for ME and Versus Arthritis, but also Remember and the Sussex ME CFS Society and sedsconnective.org. And if you have any queries for your GP about hypermobility, get them to check out the EDS UK GP Toolkit. And if you want to read more about the work that we've been doing, please look at me on Linktree, Bendy Brain. Thank you.
Thanks so much, Jess. It was very good. I think we have some minutes now for questions to either Simon or Jessica, or comments. Alan, was anyone on the chat?
Yes, there was a question on the chat from Helen Baxter from 25% ME group, very much to the Simon, which was, what do you do about PPI for severe ME as they're too ill to participate? And if I can jump in with a couple of questions as my own as well. One was, I did notice that he ended up with very small numbers of participants in the trials, given that we heard from Karen that 100 wasn't enough for a clinically significant result. I wondered if trials of 5 or 6 generated significant results, or results that could be taken forward.
Okay, Simon, so 2 questions actually for you.
Yeah, so Alan, the question about the 5, that was for a research study, not a clinical trial. The Light Me Up is a feasibility trial, and that is 10 participants, and that's basically to assess the safety, use and compliance of the lamps. Question regarding how do we involve
PPI.
severely affected patients in PPI groups. That is through principally communicating via their carer. So we have 2 carers on our ME PPI who are looking after severely affected patients, so they communicate through their carer. And also they also contribute via social media, via emails, designated emails, etc. So we take advantage of that as well whenever we can. Was there another question I missed?
No, I think those are the only ones on the chat.
Thanks.
Anyone with a comment or a question?
We have Graham, Edis and Matt Rowley. Oh,
Graham.
Hi. I was going to leave this until later at the end of the whole session, but the idea that Professor Carding might miss this troubled me slightly, because his information has been really links into what I wanted to say very, very closely. Thank you all to the previous speakers. Everything's been very, very interesting. I'm a CFS ME sufferer. After 3 decades of searching and experimenting, I found a method of treating my CFS ME, which I think is worthy of further investigation by qualified medical researchers. And I think there's already some encouraging research in these areas, but not in the combination I'm using it. I'd dearly love to pass the information on for further research, particularly as my treatment is widely available and inexpensive. For those of us that love numbers, using the mental fatigue scale questionnaire, my score improved by 37% between January last year and March this year. In short, I was a computer programmer who by December 22 was struggling to log into a computer and was housebound. I'm now able to write programs again and drive short distances. My case history is simple and points to a damaged microbiome as being the cause of my illness. Monica Bolton advised me that the treatment method I'm using may be of interest to Professor Carding and others at the Quadram Institute, because the treatment is closely linked to micronutrients, the microbiome, FMT, pre and probiotics, diet and nutrition. Although the products I'm using are available to the general public, I don't want to go into details of my treatments here, as there are dangers in what I'm doing, which is why I've consulted a very experienced doctor of food science and nutrition for guidance and have been very careful with dosage. And I've also taken extensive notes of my progress over the last 18 months. I think that what I've found may provide some insight into the interaction between microbes, gut health, and nutrition in relation to CFS-ME. After many months of searching medical research papers around the subject, I seem to have reached the limits of research knowledge in two associated directions. The information I have may also be of benefit to sufferers of IBS and IBD, and maybe even long COVID. For me, it resolves gut motility problems and has solved some of the earliest symptoms of CFS illness that I had, which implies to me that I'm addressing the core problems rather than the cascade of secondary symptoms that followed from the cause of the illness.
In the interest of time, Graham, I apologize to interrupt. I'm sure we can link you up with Simon. Is there any particular question or comment that you have? Because otherwise we can't let the others have their comment or question.
Sure. No, linking up would be absolutely fantastic, and thank you very much.
Oh, that's brilliant. Thank you so much. It's really interesting. This microbiome is all the rage at the moment. I think there was a question from Matt and one from Sarah. Matt, go first, then, please.
Hi. Well, I guess the first question is around the red light therapy. I'm sure my wife has done stuff with red light therapy, like in beauty, in a beauty context for acne and things like that. Is that the same thing?
No,
that's being looked at here?
Red light therapy is light of a specific wavelength. So like you may go down, to your lamp store and buy, they have a wide range of wavelengths, some of which can be beneficial, some can be harmful. So no, the lights we have specifically designed to emit light of a specific wavelength.
Right.
Okay.
Sarah,
so
we have room for you for a final question from Sarah. Go ahead, Sarah.
It's not a question as much as a comment, and please forgive me if I'm telling you how to suck eggs. The work I've done with physios for ME and from the studies that I've done, we've been inundated with participants when we've gone directly to them and recruited via social media and support groups and the like, rather than going through any medical centres or any medical route or healthcare related route, because so few people with ME are actually in contact with any kind of healthcare. We're kind of sorting it out for ourselves because most of us avoid healthcare professionals if we can. I speak as a healthcare professional, so I suspect you're doing this anyway, but that was the way that we've done it very successfully.
Can I reiterate that, Sarah, that we've in our studies, we have generally found it much easier to recruit from support groups and from bulletin boards and social media than we ever have through the NHS.
Absolutely, yeah. I have to say, it's not only in ME. I found that in other conditions as well.
No, these are a variety of other conditions, and we've
helpful tips. Thank you so much. Okay, I think we're coming to the second short break, 10-minute break. I think we'll come back at 10 minutes to 3. No, am I saying this wrong? We'll come back at 3 o'clock. Sorry, I got confused. And then Professor Vinod Patel will start his presentation. Can I thank the speakers for this wonderful second bit of our workshop and everybody for participating so lively. We'll get back in 10 minutes, all right? Monica, okay? Alan?
Yeah, as I understand it, that's correct. I mean, I did have one quick question for Simon again, if he didn't mind, which was, did he actually have a definition of severe ME and very severe ME that he used to recruit people?
Simon? I might have already left.
Yeah, sorry, Alan. Yes,
here
we
he is.
did.
Yep,
We,
yep.
we relied on our clinical colleagues there, and it's based on the Canadian consensus criteria. But essentially, very severe were those patients that were bedbound and in a very controlled environment with little exposure to light and sound. And the less severe were those that were able to have some ability to move around the house, not be entirely bedbound.
That's helpful. Thank you.
Thank you. We'll come back at 3. All right. Bye. Yeah, very good. It didn't pick that up. Okay, let's see whether Professor Vinod Patel is with us. Give him 1 or 2 minutes.
Helen Baxter has offered to phone him, and I assume she's doing so. So hopefully we'll get some feedback from Helen.
If anyone has any urgent comments or things that they want to address, they can do so now while we wait for Vinod Patel.
Can I just ask if it's possible? I just wondered, because obviously it seems to me like a lost opportunity, but I was just wondering, as a sort of person who's lived with ME, chronic fatigue and various things around that, if there were any trials, is it not possible, whilst we are here, that there could be some way that we could put ourselves forward? Just because I rarely seem to be in the loop for anything.
I think if you can send an email to Monica, we'll keep your details.
Brilliant, perfect.
Not a problem.
Thank you.
I think Matt's had his hand up.
Hi, yeah, it's just kind of just it's probably a bit of a left field thing, but I thought I'd mention it. I've been experimenting with different cocktails of supplements over the last few months to see if anything helps, and I think it's something called L-tyrosine that's really been helping me. I'm not obviously suggesting anyone should use it, but it was just a comment that it seems to be really helping with brain fog in particular.
Thanks, Matt.
Sorry, Matt, what was that supplement?
L-tyrosine.
Thanks.
Okay, brilliant. Does anyone know whether Vinod Patel will be joining us? Or?
He is due to join, but he was in a meeting until 3, so I'm hoping. So I suggest we carry on with discussion, and then when he does arrive, we can go back to him.
Monica, it's Helen. Yeah. Can I just say he's just logging in now?
Oh, fantastic.
I don't see any hands left. Do you,
Alan? Ewan.
Yeah, can I just jump in? Thing that Jessica's talk would sort of made me wonder. With younger people now sort of having been possibly tested at birth, are we seeing any cohorts of people with genetic backgrounds? You know, because obviously most of our people, we don't get them genetically tested until they are patients participating. But so we have nothing to compare their current state to. And I'm just wondering, you know, if we've got enough data. In the...
This might be something for the biobank people that we've been in touch with, Monica.
There's a board in Bradford study that I really want us to get some pressure on, because they're doing genetic studies. They have no interest in fatigue at the moment. So we need to start trying to get them to be interested. I don't know how we go around doing that.
Well, I know several of them quite well, actually. So happy to link up with them. Yeah.
Thank you.
All right. Let's see. Vinod, are you online?
It's Helen. Dr. Charles hasn't got a link. Alan, can you send him a current link, please?
I sent him one earlier, a few minutes ago. Well, not less than 2 hours ago.
He's got a link to Teams.
Oh, it's on Zoom, it's not on Teams.
Yes. Who's it from, Monica, please? And has it gone to the Warwick email address?
Yes, it went to Warwick and it was on Zoom.
It's gone to your...
And I sent it to 11:22.
I'm sending another one now.
Can I just, while Dr. Hal is doing that, can I caution people, don't rush out on things like LDN, vitamins, whatever. The placebo effect is a very real effect. You know, 35% placebo effect means a lot of us respond on whatever treatments. We really should be waiting for the clinical trials if we possibly can, because it costs a lot of money to try these individual treatments. I'm not going to say don't try them, but there are risks as well, that some of these drugs have side effects which you may or may not know about. People who
the good thing about the placebo effect, Monica, is that there's a placebo effect, so you might benefit.
That's true. That's true. And actually, from that point of view alone, you should be trying them. But if you're on opiates, for instance, LDN is not recommended at all. So just be cautious rather than just rushing into things that you're hearing about today.
Helen, I've just sent you the link because I don't have Vinod Patel's email address on my email system. I'd have to go up right into Eventbrite. So when it arrives, if you can just forward it to Professor Patel. Ah, here he is. Here he is. Good afternoon, Professor Patel. Just connecting to audio here. I will need to make Professor Patel a co-host, and that means I have to find where he's disappeared to on my screen.
Alan, I don't think he's got a presentation.
Right, well, if she hasn't got a presentation, then I don't need to make him a co-host.
Okay. Are you with us, Vinod?
You need to unmute
Yes.
yourself.
Yeah, I've unmuted myself. Yeah.
Okay, that's good. We can hear you.
We have
Brilliant.
you. That's
Okay.
really good.
Right. Hello. Hi, everybody. Hi, Harm. And Helen, thank you very much for putting me up for this. I think Monica had something to do with it as well. So I've got a few minutes really to talk about my small amount of experience with low dose naltrexone, but also what I'm particularly interested in is other people's perspectives to see whether this is something that we should expand, because our experiences have been variable, really. So as you're all aware, so Harm, is it all right if I just talk, or do you want to ask me questions, or?
No, you start. Go ahead, Vinod.
Yeah, so let me introduce the topic. So basically, there are very few drugs, as you're aware, for chronic fatigue syndrome / ME. And one of the treatments that a lot of people have been quite excited about is low dose naltrexone. And the usual dose that people would use is actually not commercially available in England. Well, certainly not to our local pharmacists. So we tend to use a 50 milligram tablet, which is widely available, and then you have to chop it several times. So you have a 50 milligram tablet, you chop it into eighths, and then take half of that, which is about 3 milligrams twice a day. So that in itself is difficult. How does low dose naltrexone work? Well, what it's supposed to do is that it's an antagonist at opiate receptors centrally, and it's thought to there's thought to be opiate receptors that are overstimulated centrally, or the endogenous opiate in the CFS, CFS fluid is creating activation of inverted commas the wrong pathways, and it does actually help some patients improve wakefulness. Otherwise, they think that without this drug, I'd be in a wheelchair. We do a colder tiredness score before and after, and we've started to collect some data. But within patients, we've got the ones that definitely want to continue with it show an improvement, but we don't know whether that's an accurate way of capturing improvement or not. So I think I'll stop there, and I'm sure that this exercise would be better if you ask me as many questions as you want in the small amount of time that we have. Yeah.
Well, this was particularly, I think, about your UK clinical experience. Do you have any particular lessons you've learned so far in prescribing this drug?
I mean, the main lesson is that we will try it in our somewhat more desperate patients, or for people who actually ask to go onto this treatment, and then we'll offer it to them via one particular person in our department who is then collating all the data for the patient. It's early days yet, but availability of the drug is certainly very good in our service, and at least the 8 to 10 patients that have continued on it report good outcomes. So it is quite positive in that way. So I'm just wondering what other people's experience is, or which direction people think we should be going with this, really. But we hope to have some, data on about 20 patients to put into an abstract or a presentation in due course.
Thanks, Vinod. There are 3 hands I see. I think Ewan was the first one. Go ahead, Ewan.
Right, yeah. There were originally about 6 different patient groups which were being targeted with low-dose naltrexone. Are you looking at any other patient cohorts at the moment, or have you looked at any? Or do you know of any progress that's been made in other patient cohorts for defining who benefits?
You mean in terms of within the cohort of patients with chronic fatigue syndrome, what subtypes?
No, the MS subgroup and the I can't remember what the pregnancy issues were that were.
Well, when we looked at the review, because we had to take this to Drugs and Therapeutics to get approval for local prescribing, then we didn't find any compelling evidence in any condition, to be honest, including chronic fatigue syndrome. There are several case studies, and I think the largest case study only had about 6 or 7 patients on it. But so we're just gathering our own sort of evidence slowly but surely, having resurrected our clinics under a slightly different format. Yeah, we may also think about this for long COVID, by the way, because we've taken over the long COVID service as well, because the respiratory people and ourselves now do it together. So that might be another indication of long COVID. Yeah.
Thank you so much. Matt, you're the next one.
Hi, I was just, I don't know if this is a question or a comment really, but it was just to point out that as people might know, that the CFS lot are a desperate bunch with so few options out there. And I've only recently joined a CFS subreddit, and a lot of people use LDN to what's seemingly good effect. It's actually what got me interested in LDN in the first place, and I think actually where I spotted this this discussion, which is encouraging. But I just thought to point out that there is seemingly a lot in the community which are managing to get hold of LDN and seeming to have a good response to it. So.
And I think we probably maybe through this mechanism here today, going forward, is maybe I don't do much research in chronic fatigue syndrome itself, having semi-retired. Then we should think about a protocol that we can set up, so we can at least collate some data on just a before 6 weeks and 3 months or whatever, just so that we can have some science behind its usage, really. Because obviously, if it works, we need to roll it out a lot more. If it doesn't work, we shouldn't be using it.
Yeah, I mean, as far as I know, there's only Dixon Pharmacy, which actually you can kind of get it within the UK. So I'm not sure it's just a case of asking them for their data or not, whether it might be something they'd be interested in sharing.
Would they have data on patient audit though, in terms of cold?
So I've not obtained it yet, but you need proof of diagnosis for one of a number of things. So I can't remember what they'll prescribe it for, but it's fibromyalgia. I think it's fibromyalgia, CFS, and a few other things they'll prescribe it for. So they should have a record of who they're prescribing it to and why. So whether that's so there is some
Dixon Pharmacy. Yeah,
I think it's Dixon Pharmacy in Glasgow. Yeah.
Monica's shaking her head.
I have spent many years talking to Dixon's Pharmacy. They do have data. They don't have any objective data. When I say objective, they don't have any longitudinal data on whether people have improved or not. They are indeed the main dispensing chemists for the whole of the UK, including England. So if you have a GP who is very kind and will give you LDN, the GP's prescription actually goes up to Scotland and then to a secret pharmacist down in England who prescribes it. Don't ask me how that works, but that's the arrangement they've got at the moment. There are other places in the UK. There are a lot of other places. There are a lot of private prescribers of it. They are far more expensive than Dixon's chemists. So in general I'd warn against. At least Dixon's chemists have a method of a doctor will interview you. They don't just hand it out, and they prefer a diagnosis. They will give it for chronic fatigue syndrome. I've had telephone conversations with them as a patient occasionally. But what I actually want to ask Professor Patel is, you say you got local prescribing permission. How difficult was that? Because that is a big barrier for people for other clinics in the country.
Yeah, I think what persuaded it locally was we had a good presentation from the registrar in terms of the oral presentation and the papers that they presented, but also it's quite cheap because we use a sharp knife rather than the Dixon supply that costs £50 a month, really. So that's been helpful as well. Our costs work out to about £4 to £7 a month or something like that, but it is sharp knife based rather than anything more than that.
And can I ask another one? What measurements are you taking? And are you doing any bloods?
Bloods we would have done anyway. It'd be interesting to know what recommendations you have about the bloods, because we're quite bloodthirsty in our service. We do lots of bloods because we're endocrinologists, so we're obsessed about thyroids and livers and kidneys and whatnot. So what bloods would you recommend? Because I don't know, or not recommend, but what sort of bloods improve or not improve or monitoring?
And basic ESR is an interesting one, and a sensitive CRP, because
which we don't have. That's the problem. We don't have the highly sensitive CRP. We have the standard CRP, which is only good for anything bigger than a tooth abscess, essentially.
And the inflammatory markers. I mean, there's the paper by Younger, which has got various
cytokines, you mean?
Yeah, cytokines. That's the word.
IL-6, IL-4, yeah.
Yeah.
Okay.
Yeah, we don't measure them. See,
No.
we're clinical, unfortunately, and not. I mean, we set up the service purely as help for patients.
Right.
We've
Thanks.
Yeah.
I think there's another question, Monica, if that's okay, from Ewan. Ewan, go ahead.
Yeah, I'm just going to raise early on, Dixon's, and Monica may remember, but I forgot the name of the doctor that was involved originally. They found a difference between cutting up pills and using the suspension. And, you know, the suspension was much better, not just in tolerance initial.
That's interesting.
But also in results, although your results were still patchy.
Can I ask you though, what was the dose that they use? Because the dose that we usually use, and if anybody's got experience on this, would be great for, is 3 milligrams BD is what we aim for.
I think it's a range up to 6, and you stage up from 1. Monica may probably remember the details better.
Is it 6 daily or 6 BD or?
I think be daily, yeah.
6.
Garen
That'd be 3.
has a lot of experience with the dosing. Garen, what do you think?
Oh, I just had to unmute myself. Well, I think that I did some of the same stuff as you are doing now. I started getting some experience with the LEN, but I used the titration approach in the beginning, titrating slowly up, and I dosed it only once a day, and I went up to 4.5 milligram at the beginning, and then I have tried higher doses.
What was the highest dose that you used?
9 milligram at
how much?
9.
9 once a day.
Yeah, and I've also tried dosing 2 times a day in some selected patients, but I did not see any particularly advances in doing that. But
That's a good point to for us to
we
consider as
don't
well.
know why should we dose it once a day. I think it depends on the mechanism, which we don't know for sure.
I don't know what the half-life is, but when naltrexone is used for
It's
other
13 hours the half-life, so you cannot have steady state. So the dosing once a day is based on the theory that it boosts the endorphin system, but if it's another mechanism like an anti-inflammatory effect, maybe it would be better to dose twice.
I think the half-life I thought was 6 to 8 hours or something. So if it's that, then maybe it should be twice a day. I don't know. Yeah.
Yeah, it has an active metabolite, but I don't know how that metabolite works as well. So
Plenty of questions. Anyone else with another question or another? Juan, is it something you wanted to say?
No, well, it's just for your information that the company or the lab, Accord Healthcare, is producing LDN. It's producing LDN at 1.5, 3, and 4.5 milligrams for the trials in Crohn's disease. And they are the one. This company is the one that provided us the tablets for trial. So we didn't have to prepare anything to prepare the tablets. That made the things very easy for us.
Yeah, yeah.
So you might want to get in touch with Juan Vinod to see which company he uses if you want to.
Yeah. So if somebody could maybe, whoever contacted me by email, I think it's Monica, wasn't it? Then I can have a chat with Juan.
I've got there are issues with importing drugs, unfortunately. I know that we were sourcing a source of LDN for the potential clinical trial we were doing, and the particular facility that was manufacturing in the UK lost their license for a drug standard, for a standard of drug for a clinical trial, which is higher than the standard of drug for normal use. And that the MHRA said that they would not allow importing of drugs. But I'm sure that we should try again. So if you could put your details of the drug company, that would be really helpful for us.
Monica, Juan has already put it in the chat.
Oh, good.
Accord Healthcare.
And just the other quickie, Nancy Klimas, who is quite an experienced prescriber of LDN, uses up to 9 milligrams as well.
9, okay, that's important to know. And you give it once a day. I mean, certainly some people would have side effects if you give it too late at night anyway. So in the morning is probably better. Yeah.
Well, thanks, Vinod. Anyone with a final remark or question to Vinod or anything you want to add, Professor Patel, to the discussion?
No, I think it's really good to know that we're not alone out there, and there might be some progress over the years, but we probably need to tighten up the science behind it a little bit more. Yeah,
we're totally in agreement. Yeah, I think for us it's been quite a long workshop so far, at least for me, which is great. I would like to thank everybody, and particularly Monica and Alan and the team for organizing all this. It was great to be present, and I hope it was a good interaction. Any final thoughts, Alan or Monica?
There's one final question, if you're prepared to take it, from Sarah Selke. She says, studies need to be done on LDN where patients are titrated to maximum effective tolerated doses. This seems to vary quite a bit. Does anyone have any hypotheses as to why some patients will have a flare of their ME symptoms as dosage is titrated upwards? Could it be due to a very mild reduction in certain immune responses in those who have active low-level chronic infection? That's quite a long and complex question, but
I could come with a tiny little answer on this one.
Yeah, go ahead.
So it's always been our sort of, I mean, we use naltrexone and related compounds for opiate overdoses. So how can a compound that you use in high dosages for opiate overdoses actually help chronic fatigue? So it seems really odd, actually. So what we probably, the official sort of ideas out there is that if you inhibit the opiates mildly, as it were, then you'll get a feedback mechanism, then you get increased production of endogenous opiates. But then after that, then the system becomes stunned and it no longer responds. So that's our sort of vague understanding of start low, and then we go to say we only use 3 milligrams BD, but today I think I have the confidence of using a slightly higher dose. Yeah,
makes sense. All right, I hope that's an answer to you, Sarah. Okay, I want to once again to thank everybody for their wonderful contributions and the lively discussion we had. I'm afraid we haven't really solved it yet, Monica. I always hope to sort of find the final clue and say, well, now we know what next to do. It's still a very, very difficult puzzle from many sides, but we'll just go on and keep trying.
But we have friends in the LDL world, so that's really good.
Yeah.
All right. Cheers.
Monica.
And thank you very much. Thank
Thanks.
you.
All right.
I'll leave. Thank you very much. Cheers.
Bye.
Take care.
Bye.
Thanks, everybody, and thanks so much to Harm for chairing it.
Thank you, Harm.
My pleasure. Thanks. Bye.
Thanks, everyone. Thank you.
Bye, Val. Bye, man.
Bye.